All right, so today we're going to talk about something pretty incredible. Imagine the biggest, most massive filing cabinet you can think of, one that has a folder for every single living thing on our planet. We're going to dig into the fascinating story of how we, as humans, have tried to bring some order to the beautiful, sprawling chaos of life on Earth. I mean, just stop and think about that for a second. You've got everything from the tiniest little bacterium all the way up to a giant blue whale. Millions and millions of species. So how on earth do we even start to create a system that can catalog all of them, figure out how they're related, and give every single one a unique name that works everywhere? It's a huge, huge challenge, but it's one scientists have been chipping away at for centuries. And to tackle that challenge, a whole new field of science had to be born. You know, it really comes from a basic human need to find patterns, to create order out of what looks like chaos. And when faced with the sheer diversity of nature, scientists needed a dedicated discipline just to make sense of it all. And that discipline is called taxonomy. Now, it sounds a little formal, but it's really the science of sorting things out. But it's way more than just putting things in boxes. It's about figuring out what makes a species unique, giving it a proper scientific name, describing its features, and ultimately finding its correct drawer in that grand filing cabinet of life. So where did this all begin? Well, not surprisingly, the very first attempts to classify life were all about what was practical and easy to see. These were the first really basic filing systems for the natural world. And right away, we see this fundamental split in how you can approach this. On one hand, you've got an artificial system. Think of it like organizing your bookshelf by the color of the book covers. It looks neat and it's useful for finding a blue book, but it tells you absolutely nothing about the stories inside. A natural system, on the other hand, tries to group things by their real relationships, like sorting those same books by author or genre. That reveals much, much deeper connections. And the first major scientific crack at this came from Aristotle. His system was definitely an artificial one. He basically created two giant filing drawers, one for plants and one for animals. Simple enough. Then he sorted them based on really obvious traits. You know, does it move? Does it have red blood? Is it a big tree or a small shrub? It was a brilliant start, for sure. But it was basically like organizing that whole filing cabinet just by reading the labels you could see from across the room. So for hundreds of years, classification was kind of a mess. But then, in the 1700s, this Swedish botanist named Carolus Linnaeus came along and just completely changed the game. He's the one who gave us the foundation of the system we're still using today.
Linnaeus's true genius really came down to two incredibly powerful yet elegant ideas. And these ideas solve two of the biggest headaches in taxonomy. First, how do you organize everything into logical groups? And second, how do you give every species a name that makes sense and that everyone can use?
His first brilliant move was binomial nomenclature. It's basically a two-name system for every living thing. You can think of it like a person's first and last name. You have the genus, which is kind of like your family name. It connects you to your close relatives. And then you have a specific epithet, which is unique to you. For us, that's Homo sapiens. This simple, brilliant idea created a universal language for all of biology. And this is exactly why we needed that system so badly. Common names are a total disaster. A jellyfish isn't a fish at all. A crayfish is a crustacean, also not a fish. A silverfish, that's an insect. And a starfish is an echinoderm, definitely not a fish. You see the problem, right? It's just total chaos. Scientific names slice right through all that confusion. And the beauty of it is just how simple the rules are. They're universal, it's always two parts, always based in Latin, the genus gets a capital letter, the species is all lowercase, and you always write it in italics. It's this simple, elegant code that just works for every single species everywhere in the world. So Linnaeus gave us this amazing structure, right? But a new scientific revolution was brewing that was about to completely change the purpose of that structure. The next big leap wasn't just about organizing the filing cabinet anymore. It was about understanding how that cabinet was built over millions and millions of years. And this is the absolute key. After Charles Darwin published his theory of evolution, taxonomy was transformed. The goal wasn't just to sort and label things anymore. The new goal was to create a classification system that actually reflected the real evolutionary family tree of life, what scientists call its phylogeny. And you can literally see this shift in thinking right here on this timeline. We started with Linnaeus and his two simple kingdoms. Then, as microscopes got better and revealed this hidden world of tiny single-celled things, the Protista kingdom was added. A century later, scientists realized that fungi and bacteria were completely different from everything else, so that gave us 5 kingdoms. But then,
An even bigger discovery was waiting just around the corner. And that brings us right into the modern era. The ability to sequence DNA gave us a tool that was more powerful than any microscope could ever be. For the first time, we could read the actual blueprint of life itself and see relationships that were totally invisible to the naked eye. So, in the 1970s, this scientist named Carl Woese was studying the genetics of what everyone thought were just simple bacteria, things without a nucleus called prokaryotes. And what he found was, well, it was shocking. He discovered they weren't one group at all. They were two totally separate domains of life, bacteria and archaea, and they were as different from each other as either of them was from us. It was an incredible discovery that completely redrew the very base of the tree of life. So today, taxonomists aren't just looking at feathers or fins anymore. They're looking at the code of life itself. By comparing the sequences in DNA, RNA, and proteins, they can map out evolutionary relationships with mind-blowing precision. This is how we're finally able to build a truly natural system of classification, one that's based on the actual history of life on this planet. Okay, so with all this complex genetic data out there, how does a scientist actually figure out what an organism is when they find it in the wild? Well, one of the most clever and common tools is something called a dichotomous key. It's kind of like a choose-your-own-adventure book for biology, breaking down a really complex identification into a series of simple, paired choices. Let's just walk through a super simple example. Say we find some unknown insect. The first question in our key might be really straightforward. Does it have wings? If we say no, the key might point us towards something like a silverfish. But let's say our insect does have wings. So we go on to the next question. All right, next question. Does it have a proboscis? You know, that long straw-like mouth part for drinking nectar? If the answer is yes, boom, we've got a butterfly. If the answer is no, our key tells us we're looking at a beetle. And just like that, by following this simple yes or no path, we get a solid identification. It's a really elegant tool. So all of this organization fits neatly into the modern hierarchy of life. Think of it again like those nested folders in our grand filing cabinet. You start with the absolute broadest category, domain, and then you get more and more specific, from kingdom to phylum, class, order, family, genus, and finally all the way down to a single unique species. With every step you take down that list, the organisms in that group have more and more in common. So our understanding of life's filing system has come a long, long way. From Aristotle's two simple drawers to this incredibly complex, interconnected tree based on genetic history. And the best part is, it's a story that's still being written. What new forms of life might we find in the deepest parts of the ocean or in the most extreme environments? We just don't know. Every new species we find and every genome we sequence adds another amazing page to the great story of life on Earth.